Hello everyone. As a continuation of our last class, today I am going to discuss about the antigenic structure of streptococcus pneumoniae. So the antigenic structure of any pathogenic organism mainly determines its degree of pathogenicity. So in the streptococcus pneumoniae, so many antigenic structures are there. So the most important one, capsular antigen, somatic antigen, toxins, and some enzymes are also there. So with this, the capsular antigens. The capsular antigens forms hydrophilic gels on the surface of the microorganism. So the antigenicity produces immunity, and these capsular antigens is the basics for separation of various serotypes of Streptococcus pneumoniae. So these capsular antigens may sometimes cross-react with the other bacteria due to the common polysaccharide NAG, that is N-acetyl D-glucosamine. Apart from the capsular antigen, somatic antigens are also there in this Streptococcus pneumoniae. Here, one of the somatic antigen is C polysaccharide, which is species specific. The C polysaccharide is a major cell wall structural component. The teichoic acid polymer with phosphocholine, galactosamine, glucose, phosphate, ribitol, and trideoxydiaminohexose. So the phosphocholine is responsible for the agglutination of pneumococci, and you know well the choline is an important constituent in the growth medium of Streptococcus pneumoniae, that is the Reinhardt infusion agar. This C polysaccharide binds with the C reactive protein to activate the complement mediated phagocytosis. So the next important somatic antigen is F or Forestman antigen, and then another one is M protein. So the F or Forestman antigen is a lipotrichoic acid consisting of C polysaccharide covalently linked to lipids. It inhibits the N-acetyl L-alanine amidase. And the next important antigenic structure which comes under somatic antigens are M protein. The next important antigenic factor is the polysaccharide capsule. So far 91 types of Streptococcus pneumoniae were identified based on its polysaccharide capsule. And this polysaccharide capsule has the antiphagocytic ability by inhibiting the C3b oxidation process of the bacterial cells. And the next one, the choline binding protein A, pneumococcal surface protein A, that are adhesins, which facilitates the attachment to the mucosal surface by attaching with N-acetyl galactose. And another one important group of antigenic factors are enzymes. So under enzymes, two important enzymes are there. One is neuraminidase and another one is protease. So the neuraminidase here cleaves the terminal N-acetyl neuraminic acid, which invades the nasopharynx of host cell. And then the next one is the proteases. Proteases degrades the protein molecules like antibodies, IgG, IgM, and secretory IgA. So another important antigenic structure, that is toxins. They are all one of the most important determinants of pathogenicity. So these organisms produce mainly two types of toxins. One is pneumolysine O and another one is autolysin. So pneumolysine is a 53 kilodalton protein that is cytolytic to the eukaryotic cells like human beings. And these pneumolysine have cholesterol as a component of their cell membranes, particularly the respiratory epithelium. It also activates the complement. Another important toxin is autolysin. This autolysin causes the lysis of pneumococci in the presence of surface acting agents or antimicrobials that inhibits the cell wall synthesis. And it releases toxic proteases. So the cell wall components like teichoic acid and peptidoglycan are also playing a role of antigenic structure. The next important thing is diagnosing a particular pathogenic organism. So the diagnosis of Streptococcus pneumoniae mainly involves direct examination of sample. Here it is a sputum sample. So we can directly gram stain that sputum sample, and then we can go for microscopic observation. And for culture, we are going to use the Reinhardt infusion agar. So the appearance of alpha hemolytic colonies that are also bile soluble and oxygen sensitive, and the strain which will also show positive Quellen reaction is found to be Streptococcus pneumoniae. If the typing sera is available, this method, that is the Quellen reaction, is the most simplest, rapid and accurate test. Pneumonia. So another laboratory diagnosis is also there. That one is serological diagnosis. That is the detection of pneumococcal antibodies simply by radioimmunoassay, and capsular polysaccharide detection can also be done by simple counter immunoelectrophoresis. So how can we control the Streptococcus pneumoniae infection? By means of chemotherapy, that is by giving antimicrobial agents. So based on the sensitivity testing, so various chemical agents are used to treat Streptococcus pneumoniae infections. The main thing is cephalosporin or erythromycin, chloramphenicol and quinolones. So most of the quinolone drugs are used in treatment of Streptococcus pneumoniae. So the pneumococcal conjugate vaccine can be used for very high risk cases, and moreover pneumococcal vaccines are available. So in the year 1977, 14-valent polysaccharide vaccines were licensed, and in the year 1983, this 23-valent polysaccharide vaccines were licensed. In 2000, 7-valent polysaccharide conjugates were licensed. Nowadays, so many pneumococcal polysaccharide vaccines are available. So purified capsular polysaccharide antigen from 23 types of pneumococcus are available. So it accounts for 88% of bacteremic pneumococcal disease. It can cross-react with types causing additional 8% of disease. Nowadays, children under the age of 2 are given the pneumococcal vaccine by the pediatricians. So with this, the Streptococcus pneumoniae portion is over for you, and I will meet you with the next organism. Thank you.